But
Welcome to the Bio and Beyond podcast. I'm your host, Satwik Nath, and today we're diving into the world of biochemistry with our distinguished guest, Christian Choe. He is pursuing his PhD from the Stanford Department of Biochemistry, and he has worked on a number of research areas related to RNA and proteins. He has also published research papers in top journals. Join us as we explore the fascinating intersection of biology and chemistry with Christian on today's podcast. So why don't we start off with a simple question? Uh, how did you get into biochemistry? Let us know a little bit about you.
so I guess I first got into sort of science in high school. I was really into chemistry and like I ended up getting involved in like science Olympiad, chemistry Olympiad. And then in high school, that's when I first actually tried to do research. So what I did was I actually just cold emailed several professors like at a local college to try to do chemistry research. So as I was able to do a little bit of chemistry research in high school, and then in college, I got into chemistry a little bit more, going into like chemical engineering. But I feel like in research, I was always more interested in biology. So then what ended up happening was um, I got in like my undergrad, I did more sort of biochemistry research or bioengineering research. And then that ended up leading me to doing sort of more protein and RNA research now as a PhD student. Um, my path was very non-linear, but I do think from my takeaway from doing this was a lot of times your research interests sort of change as you learn more. And from my experience, it's important to just keep exploring other options and don't try to settle too quickly, especially when you're not certain of what you really enjoy.
Well, that's really inspiring because originally you didn't want to do what you're doing now.
Yeah, originally I was actually like in high school, I was really obsessed with just like organic chemistry. And
Hmm.
I was like, oh, I really wanted to get into like research related to like total synthesis of like, oh, can I like medicinal chemistry, like how do you use chemistry to make, new drugs or like new therapeutics. And that was sort of what I was really interested in high school. But then in college, I was more exposed to, I would say, biochemistry broadly. And like I learned a lot more about sort of biology. And honestly, I think to me, biology is really interesting. Like all of life that we know is all controlled by biology. And like in some ways, all of human suffering is related to biology. Like, and sort of just looking at biology from all these different perspectives, it's really amazing, like how it can function and how it can work. And that sort of led me to be more interested in like sort of proteins and RNAs because in, from my perspective, like DNA, proteins, RNAs, and all these small molecules, they're all the language of life. Like how a cell communicates with each other, or like how a single cell can survive in its environment. Like these are all different molecules that sort of represents like how it can even do all of these crazy functions. And that sort of led me down the rabbit hole of like biochemistry, which is how I ended up here.
Oh, that's really interesting because that's different than the path that most people take. I know a lot of people who know what they want to do in high
Hmm.
school and like go straight to that. But you obviously chose to do something else in high school. You wanted to do something else. And then when you got to college, it was completely different. So, how would you say that has impacted you differently from other people?
I guess the one thing that could be a pro or con about this was it made my life a little bit more hectic. Like in college, I ended up when you switch your interests and like you end up taking you have to, take more classes in other fields that you're lacking in. So I guess I had more catching up to do by changing what my interests were. But I think it helped me get a broader sense of maybe the field in general. So I feel confident with my background in chemistry, in biochemistry. And from honestly, I do think science is very interdisciplinary. So like even if you're interested, like let's say in high school, you're really into like biology, and then in college, you're like, oh wow, I really like computer science. There are many intersections of like biology and computer science. And the classic example is like bioinformatics. But honestly, like research is hard to put in a box because you could argue that there's so many interdisciplinary fields of research that they don't neatly fit in like a single major or like two majors. Sometimes they do sort of the boundaries of it are a little ambiguous. So I think that's more interest. The more important thing when you're pursuing your research interest is more your personal interest because that is hard to define like within a single major. But of course, you do have to pick a major in college, and I think that should be broadly what you think best fits you as a person.
Okay, so now that we spoke about how you got into what you're doing currently, why don't you tell us a little bit about your journey as a researcher?
Okay. So like as a PhD student, I would say the start of it was a little bit rough. Like I have had many experiments fail, and sometimes you do feel like as an early scientist, you don't want to tie your self-worth to your research because you'll always have experimental failures, and but those are always things that you should try to learn from instead of viewing them as sort of a mark on yourself. Like you should try to not tie your self-worth to your own research. So at the start of my PhD, I worked a lot on like protein design, trying to design sort of like different protein-protein interactions to make like a protein interaction network. And then later on, when COVID happened, my research shifted a little bit towards like the COVID mRNA vaccine stabilization. And then like as of now, recently we're trying to publish a paper on trying to design molecular calculators. And just to give a little bit of info on that, the basic idea is that like for like a calculator, you put in, numbers in a calculator and you can add them together. In a sense, we're trying to make a nucleic acid function like a calculator. But instead of the inputs being numbers, the inputs to a nucleic acid calculator are like different short fragments of different RNAs. And then the output for our calculator is just fluorescence. So it's sort of like a binary signal. So like zero would be off, and then one would be fluorescence. And the goal of these calculators is to sort of act like molecular sensors. So like, for example, in our paper, our use case that we use as an example is like tuberculosis diagnostics. So in tuberculosis, there are three different molecules, A, B, and C. And if you calculate A times B divided by C squared, you can diagnose whether or not a patient has active tuberculosis. And we try to see if we can design a molecule to calculate A times B over C squared.
That's really interesting. And it also seems super complicated.
It does seem complicated, but I think one thing that's important about the paper that sort of simplifies things is that the power of biology or the power of computing in general is sort of related to the number of interacting components. So like for example, if you have a system just like A and B, and those are the only two things you have in your system, then the system is very simple. Usually like will bind B or A will not interact with B. It's sort of binary. But when you start to add more components to your system, like let's say you have like n different molecules, you have potentially n factorial interactions, right? And the interesting thing about that is that every single molecule, your entire system can sort of represent a complex function. So like one way to think about like a bacteria is to think about a bacteria as a function with like 1,000 variables or maybe even several thousand variables. In some ways, like I think it's really interesting to just think about biology in the terms of like the individual interactions because like DNA, proteins, and like all your RNA, they all interact with each other in very complex ways. And because of that, biology is very flexible, right? You can get very different outputs depending on the different environmental conditions. And it's sort of the way that biology is able to be so flexible is by having like the sheer number of interactions.
That's that seems really cool. So why don't we move on to some other types of research you've also worked on? So I was reading your work and I saw that you were working on compact RNA sensors for increasingly complex functions of multiple inputs. Can you
Mhm.
tell us some about that?
Yeah. So I guess what I just talked about was about that paper.
Mm-hmm.
Um, the other thing I can add is the paper was um the goal of the paper was just to sort of see the limits of what we could try to design. So in
Mm,
the paper, we were designing sort of short RNA calculators. And to give you a sense of scale, these are like 85 nucleotides. So they're very short, small RNAs. And we were interested in just seeing like what is feasible. And the interesting thing about the paper is we involved community science. So in Riju Das's lab, we have a game called Eterna, where players around the world can actually design RNAs, and some of these designs actually do get experimentally tested. So I think currently we have challenges for trying to design just interesting RNA shapes, but during for the paper, we asked players around the world to try to design like different molecular calculators. Like we would start off very simple problems, like, oh, can you just make like a simple calculator to detect the presence or absence of a single input? So it's like a one input calculator. And then we got more complex. We were like, oh, can we do two input calculators? Examples would be like Boolean logic gate. Like, can you make an AND gate, an OR gate, or like a XOR gate using RNA? And then that eventually led to the final challenge, which was to design the tuberculosis calculator.
that's really interesting. So, other than tuberculosis, have you used this calculator for any other diseases, or is it just tuberculosis right now?
Yeah, for the paper, we just focus on tuberculosis as the proof of concept. There are other diseases like sepsis like that have similar functions to calculate, but we haven't tested those out yet.
So, do you plan on testing those out in the future, or?
As of now, it's hard to say.
Mhm.
Um, because I think we were more so we have developed like a computational design method. So now you can sort of automate these designs. So in principle, if other people were interested in trying to design complex calculators, they can just run the program and see what they get. But in the lab, I would say we have shifted our interest in the lab. So I don't think, at least in the short term, we may not pursue sort of complex diagnostics. Though it's unclear because I do know that potentially we might be exploring sort of like trying to build like an RNA computer. So like the idea of like, you know how a calculator, the most basic calculator is basically an adder where you just add numbers together. But can you do that with the RNA? Like, can you just add numbers together and can you have like a carryover bit in RNA so that you can have like like a an RNA calculator that can have like multiple digits? So that could be that might be potentially what the lab explores in the future.
How did you find an interest for this topic? Because this sounds really interesting. And like, how did you find this topic and what made you really interested in it?
Mm, so when I first joined the lab, they were working on um, the like how do you design like the tuberculosis calculator by hand. So players have designed them, like obviously players have designed them by hand and submitted them to the Eterna game, but the question was like, was there a way to automate the design? Because sometimes like it would be convenient to automate the design of complex calculators for many different use cases, depending on like what people wanted. So that's sort of how I first got involved into it. But then I think my interest sort of grew because my professor was interested in sort of justifying the idea for this from a theoretical basis. Like, even if it's feasible to do this, like, why in theory should we do this? And one thing that's really interesting to think about is you can you can in theory have a single molecule approximate any arbitrary function. So like for example, let's say you draw out some function on like a XYZ plane, and the only caveat is we only work with the positive numbers, and your function has to just be a positive output. But if you just draw any arbitrary function, you can actually turn that into a single molecule. So any single molecule can represent any arbitrary function, which to me was really interesting. And also, it just goes to show like the power of biology, right? Like every single molecule in your body can also approximate a function. So in some ways, if you want to think about a cell, you can think about a cell as a list of like hundreds of functions that are all interconnected. And like I think that's obviously it's really hard to fully like understand every component of a cell, but I think it just helps me appreciate like the complexity that even the simplest things in biology can have.
Mm. So while we've been talking about this, you've been using a lot of math terms. Would you say that biology, like what you're working on, has a lot to do with math? Because like you've been creating calculators and you've been comparing what you've been doing to functions and all that.
Yeah, I would say, I mean, it's the nature of my PIs. So I'm co-advised by two different PIs, and both of them, one of them is very like has a physics background, and the other one's very computational. So I would say my approach to biology is maybe more physics and more math oriented than other people's. But I would argue that honestly, every every viewpoint of biology has value. So even if you approach it from an ecology perspective or maybe a more like environmental perspective, that is also that can also lead to very interesting results. Maybe it was just my background is more math and computational. Like in undergrad, I did more engineering classes a lot, so that probably has something to do with it.
Yeah, that's quite interesting. It goes back to your background again, which is apparently always like different from other people's backgrounds.
Hmm.
So, how would you say that your PIs have influenced you other than that?
Mm, I think the one thing that I find important for me in a PI is like mentorship. So like when you're starting off, like learning how to do research, it is sort of overwhelming because in some sense you know very little, right? Like there's so many papers out there and the field is constantly changing. Even when you read papers, I always feel like I don't fully understand the full scope of everything. But having a PI is nice because they sort of help ground the project, and they're also very helpful in just guiding you in like terms of what directions are worth exploring or like what directions do I need to like read into more. Um, and I find it's very nice. Um, mm, yeah, I would say in general, mentorship is really valuable in research. So like, let's say, I don't know if you've been involved in research as a high schooler right now, but honestly, like I found that all of my mentors throughout like high school, undergrad, and college have helped me grow as a scientist. Um, and honestly, everyone I've worked with have been really supportive for me. So I find I'm really grateful for that. And honestly, I don't think like I would be here without any of them.
That's really interesting how your mentors have helped you all the way throughout your journey, even from high school.
Hmm.
So, in your current research focus, what would you say you're exploring, and what do you think that the impacts of this could be in the field of biochemistry?
So currently, what I'm really interested in exploring is sort of how to better learn molecular interactions. So like for example, like like when you design an antibody against a virus, the antibody will bind, the virus with like really high specificity. And some of these interactions will be like hydrogen bonds. Some of them will be van der Waals and ionic interactions and like pi stacking. But what I'm really interested is I'm trying to train like a neural network to sort of better learn all of these molecular interactions because I'm really interested in trying to just design crazy things. Like I'm really curious, like, is it possible to design like a superconducting protein? Or like, can you design like a protein diode analogous to like a LED? And like, I think potentially you can do all of these things, but the one limiting factor for trying to design really complex proteins or really complex RNAs is it's hard for the models to fully understand like all of the molecular and physical interactions. So I'm hoping that by trying to get the model to learn all of these interactions that you can try to design really interesting proteins or really interesting RNAs with very unique properties. Um, there are use cases like you can use this to design better drugs, but I'm also interested in just seeing like can you design like crazy material? Like can you have an LED that's made out of protein? Like can you have an LED that functions in a cell? Like maybe that could be possible.
Hmm. So, do you think that these are gonna like appear anywhere in the future, like these ideas?
maybe. I don't know. So like one thing I am working on right now is sort of just playing around with sort of fluorescent proteins, and
Mm-hmm.
hopefully that will be public maybe within a year. But all of these other crazy ideas definitely will not be done anytime soon. But honestly, like the world of molecular design is never ending. Like there are so many things like that. Like I guess our knowledge right now, we can only design like a fraction of every possible thing. And if you think about biology as like exploring this molecular space of design, biology has explored like a huge area of just molecular design, but there are also areas in biology that cannot be explored by life. For example, like there are weird chemistry that you would just never see in life, but potentially that could be new avenues for crazier like reactions or like even crazier materials. um, so I don't know. I think like in some ways it's sort of like exploring space. Like space is endless. And in some ways the space of molecules is also endless. Like there are like infinite number of possible proteins and RNAs and different molecules that you can't explore them all, but potentially there is the answer to everything within that space.
Mm-hmm. So on the discussion of crazy things,
Hmm.
while working in labs, have you seen any like recent crazy breakthroughs or innovations that you were particularly impressed with?
Hmm. So I would say the most recent thing that was really impactful to my field and to a lot of other to just biology in general was AlphaFold 2. So like AlphaFold 2 was like the first instance showing that like, oh, we can build a robust like neural network model that can predict structure from sequence. And that's really impactful for a lot of people because often in biology, there is a structure function relationship. So a lot of things that have similar functions will also have similar structures, like in 3D space. But the problem is that to solve a protein structure experimentally is very difficult. And depending on the method you use, can honestly take you like several months or up to a year to do. So having this neural network that can now give you like a good estimate approximation of what a structure is has been a godsend to a lot of different areas of research. And for us, it has been very useful because when in our lab, we try to design a lot of different proteins, and having that kind of model available, we can actually use it as a way to screen a lot of our designs. Like if our designs cannot be predicted by the model, then it's possible that our designs are not very good. Of course, there are caveats, like because a neural network heavily relies on the data it's trained on. It's not perfect, but as of now, it's so much better than all the previous methods that um, it's been a really good breakthrough.
That's really interesting. So you said that that not only helps you, but it helps a lot of other fields as well.
Hmm. Like in general, I think just having an ability to predict the structure lends a lot of labs more um, power in like developing hypotheses. So like in general, a lot of like biochemistry research is like hypothesis driven. Like, you observe something in your data and then you have a hypothesis and then you want to test your hypothesis. But then I would say like, from a structural biologist perspective, it's really important to have a sense of structure in order to get a sense of like what the function is. You can guess the function based on your assays and experiments, but it's hard to understand like the molecular basis of how things are working without having like any sort of protein structure or like RNA structure.
Yeah, that's really interesting. So now that we talked about some breakthroughs that we had, what do you think are the most like pressing challenges in biochemistry?
Mm, what are the most pressing challenges? I mean, right now, I would say there are a lot of open-ended questions throughout biochemistry. Like if you're interested in like, for example, if you're interested in neuroscience, there are a lot of open-ended questions about like oligodendrocytes, which are the cells that make the myelin sheaths around neurons. Um, I know some people who are working on those kind of projects. Um, the space is sort of endless. There's a lot of focus right now on immunology because I would say a lot of new and developing therapies focus on immunotherapies for like cancers and other diseases. But I guess for me, a more fundamental thing about biochemistry that I'm really interested in is just like how do you better learn and model just interactions in general? Because if we want to design, an RNA calculator, if we want to try to design like our own life form without like that's different from biology, we really do need to understand like all of these molecular interactions a lot better.
You said designing your own life forms.
hmm
That's really interesting. Can you go into more detail about that?
So one thing that I think about sometimes is you know how every molecule in life has a handedness?
Mm-hmm.
Like you have like DNA, protein, they're all chiral, but you only ever observe one chirality. And so one thing that would be very interesting, but very difficult to do, would be could you create a chiral or like the opposite chirality life form? So like could you make a bacteria that has to feed on the different chirality glucose? And it would make the different chirality protein and RNA and DNA. And the interesting thing about this is that if you were to make a plant with the opposite chirality, then all of its cellulose wouldn't be able to be broken down as easily because fungus and bacteria don't have the proper enzymes to break down the wrong chirality cellulose. So you could imagine like trying to make a forest out of all of these flipped chiral trees as a carbon sink. Um, but then the other thing is like also like more fundamentally, what I'm interested in also is like how did life begin? Like there are many hypotheses, such as like the RNA world hypothesis, that life began with RNA. And people have shown that even though RNA, some people in high school biology, you tend to only learn RNA as like the middleman between like DNA and protein, like in the central dogma. But what's interesting is that RNA has been shown to have really interesting functions. Like RNA can be used as a catalyst. Like RNA can, there are like some exons are self-cleaving RNAs. Like RNAs can be enzymatic. So because of these properties, people think that somehow early life probably used RNA, but it's unclear exactly how you can create the first self-replicating system with RNA. So that's a really open-ended question. But like I do think as we learn more about how to design these complex systems involving proteins and RNAs, that potentially we could try to design like a very simple life form that's like a very tiny, like has like the minimal components for life. And I think that would be super cool.
Yeah, that's not really something I was expecting to hear today. Designing different life forms. That sounds like something out of sci-fi. That's crazy.
Yeah. It definitely won't be possible now or anytime soon, but I do think like as we learn more about biology in general, we'll get a better grasp about the complexity of life and how we can sort of use it to build our own life.
Mm-hmm. Well, I have one final question for you, and that's basically for the high school students aspiring to pursue biochemistry and research. What
Mhm.
advice would you have to give them?
So I would say the important thing in high school is just to explore your own interests. Like your own interests will evolve and change as you learn more. Like, especially when you start to do research, you'll learn to either love things or you'll actually dislike things. Like, I would say early on for me, when I started to do research, I initially enjoyed like doing wet lab experiments, like trying to synthesize proteins, and trying to purify them. But then later on, as I got more involved in computational experiments or computational research, I actually found that I enjoyed that much more. So then I sort of shifted my focus towards more computational studies. And I would say in high school, if possible, you should try to get involved in research. Like you can reach out to professors at like your local college and maybe also in university as an undergrad student, you should totally try to get involved in research. But also, when you do get involved in research, don't feel like you have to stick with it. You should feel free to explore because, like, oftentimes I've seen a lot of students join a lab and they feel like, oh, it's rude to leave, or like they feel like they should stick with the lab, even though sort of their interests have shifted. But I would say it's better to just Like as your interests evolve, you should also just explore other opportunities as well. And I I'm like I don't know if my example is the best example, but like from my experience and how my interests have changed throughout like high school and college, um, I would also say like don't be afraid to explore other things. Like research is very complex, and I would say research is becoming more and more interdisciplinary. So like if you feel like if you're interested in biology and chemistry, and maybe you're interested in like physics and computer science, like you can start to you should explore all of these things and see how they connect because in the end, like all of science is connected and they all need each other.
That's really interesting. And it's even more interesting that you can be like one of the examples because, of course, you didn't want to do this originally. You had other plans. And then later on, you decided to switch what you wanted to do.
Hmm.
So that's really inspiring for other high school students who may be in a similar boat, who even may not know what they want to do now.
Yeah. I mean, honestly, I feel like in high school, or like sometimes it feels like there are a lot of solved problems in science. Like in high school, when you're learning, biology, chemistry, physics, you're learning examples of a lot of things that are really well characterized and really well known. But as you go into college and you go deeper into these different subjects, you realize that there are so many open-ended questions. Like I feel like we only have touched like the surface of science, and like it's sort of endless the knowledge that exists. And I think one thing that's important though is that you shouldn't tie your self-worth to your research. So in like undergrad, I had a lot of experiments fail, and if you get too emotionally attached to your projects, you can also feel depressed because I mean science is risky and science is exploratory. So you shouldn't expect every experiment to work. And the thing you should focus on is sort of the process of doing science. Like you should make sure that when you're doing your experiments, you do them in a way that you're being careful with your controls, and you're being careful with your methodology so that even when the experiments fail, you can at least learn something from the experiment. And you should sort of, the way you should treat your own value is maybe not in your result because just because the experiment fails doesn't mean you're a bad scientist.
Yeah, I think that's also really true that you shouldn't tie yourself worth to it and that you should be more open to other options.
Yeah.
Hmm. So, do you have any final remarks you'd like to make?
Mm, well, I will say I'm biased because I think biology is amazing, but honestly, I think if you just when you start to learn more and more about biology, there are so many amazing things about biology at every level, like the molecular level, the cellular level, organismal, environmental. And I would say like just go out and explore science. Like science is endless. And honestly, like just everyone should just go out and just see what they're interested in. Like, mm, what is a good thing to say? Mm, yeah, I mean, honestly, I think everyone should just explore science on their like in their own passion. Like whatever they're passionate about, they should explore it because I think that's the best way to do science.
Okay. Well, thank you for that inspiring talk. And as we wrap up this insightful episode of the Bio and Beyond podcast, I want to extend our sincere gratitude to Christian for sharing his wealth of knowledge and expertise. His commitment to research and innovation is truly inspiring.